This method can help answer key questions in the zebrafish heart regeneration field, such as the molecular mechanisms regulating heart regeneration. The main advantage of this technique is that this method is very efficient to knock down target genes expression, and it's very convenient.
To begin this procedure, add 4.2 milliliters of the tricaine stock solution to 100 milliliters aquarium system water to prepare the tricaine working solution. Then, prepare the instruments and materials needed for the surgery, which includes a piece of sponge, a petri dish filled with tricaine working solution, a plastic pipette, a stereo microscope, 2 sharp forceps, iridectomy scissors, and elbow tweezers. Prepare a small groove in the sponge to hold the zebrafish during the ventricular resection surgery. Next, prepare a recovery tank by filling a 3-liter zebrafish tank half full with distilled water containing sea salts at a concentration of 60 milligrams per liter. Using elbow tweezers, transfer an anesthetized fish ventral side up to the sponge groove. Visually locate the margin of the beating heart, then use a stereo microscope to locate the heart. Using iridectomy scissors, begin making a small 1 mm incision that penetrates the skin and muscles. Use forceps tips to carefully tear the silvery epithelial layer of the pericardial sac to expose the heart. Make sure not to insert the forceps too deeply to avoid injuring the heart.